If you don't know where that is, it's just basically kitty corner to us. It is northwest of us in beautiful Novato, California. And Brandon was a associate of our immortal Judy Campisi. Okay, and I'm going to give this over to Brandon, and yeah, have at it. There you go. I'll put this in your pocket. Yeah.
Nothing like Greek island.
Awesome. Thank you for giving me the opportunity to talk, and thanks for being flexible on the program today. I'll be talking a bit about my thesis work in Judy Campisi's lab, talking about unique disease-relevant senescent cell states are shaped by differences in response to DNA damage. So, as you all know, this was Judy. She was a pioneer in the field of cellular senescence. I was lucky enough to join her lab back in 2020. And a lot of these projects I'll talk about today were really brainstormed with her. I was able to have like monthly meetings with her, and we came up with topics that would be what she thought and I thought would be really good for my thesis work. And that was really fundamental times in my training, and I really appreciate that times before she passed. So as Pierre showed yesterday, I started as a lab manager, a research associate, and this was the lab back in 2021. I was lucky enough to still be there when Judy threw some of her lab parties, very big social events with other labs as well. And I started my PhD back in 2022. But as Pierre mentioned, Judy's health was starting to decline, and also the lab started getting smaller. And here we see one of our probably last events we went out to with Judy before she passed. And as you know, she passed in 2024, and that was actually right before my qualifying exam. But luckily, Pierre stepped in. Judy trained us to be really independent scientists, and I really thank her for all that training because we were able to keep the lab going and help me graduate early. Within 4 years, I'll be defending next Friday. Thank you. And here's our current lab right now. We have 2 research associates and Pierre helping with the lab. And really a big thanks to our current lab and so many other labs too, and other people who have supported us to keep everything going and publish all the papers that really Judy started when I first started in the lab as well, when we were brainstorming together. And so today these are the 3 papers that we worked on early on back in 2020, and I'm trying to publish them now. We just had, as Marco mentioned, a collaboration with Rubedo. This paper detailing senescent fibroblast heterogeneity in the context of idiopathic pulmonary fibrosis and DNA damage response, and this just got accepted to NPJ Aging. The project I'll be talking about today is the second paper, looking at DNA damage drives this unique Alzheimer's disease relevant senescent state in neurons, and this is under peer review also at NPJ Aging. And then we have another one looking at cellular senescence and antiviral responses in idiopathic pulmonary fibrosis, and this was in collaboration with a former colleague in Judy's lab as well, who was here yesterday actually, Steffi, in Jena, Germany. And so all these papers we're trying to publish just to finish out the lab, finish out Judy's legacy and her publications. And so as I mentioned, I'll be discussing this paper today about a senescent state in neurons. And so to define this, I think everyone really knows, or probably knows it by this point, what cellular senescence is. But it's really this tripartite phenotype composed of this senescence-associated secretory phenotype, the SAS. This is what Judy pioneered back in 2008. The cells are also resistant to cell death. This was kind of detailed back in the 90s, 2000s. And then also the cells no longer divide. And of course, that was came from Leonard Hayflick back in the 1960s, I believe. But now cellular senescence is understood as a very complex and heterogeneous phenotype. We've seen this from a lot of talks. It's dependent on a variety of factors, not just included here, but some of the ones listed, like how the cellular senescence is induced, the type of cell type that becomes senescent. We can even see heterogeneity within the same cell type. Sometimes the temporal dynamics of how cells go to a senescent state are different, even within the same cellular cell type population. And Judy also used to always mention that cellular senescence is actually good in a variety of contexts, such as embryogenesis and wound healing. So our lab has always been interested in understanding novel senescent states in the context of disease. And this is really important to discover better therapies and senolytics. And so what I wanted to investigate and what we came up with when we were brainstorming, me and Judy, back in 2020 and 2021, was looking at senescence in the context of Alzheimer's disease. Because there's this emerging topic, as you know, neurons are postmitotic. They don't divide later in life. And so this was thought before, maybe back in like 10 years ago, that neurons were actually incapable of undergoing senescence, because one of the canonical definitions of, or part of the tripartite phenotypes, was the cell cycle arrest. So how does a cell become senescent if it's already cell cycle arrested? Around, I think, 2012, out of the Mayo Clinic, Diana Jerk and her group posted a paper about neuronal senescence in older mice, having a link to DNA damage response and P21 response. And this kind of got the field moving that this tripartite phenotype is not a defined definition, and it kind of can be fluid, and of course, cellular senescence is just becoming more and more complex with all these discoveries. What got me interested in this actually was looking at the canonical inducers of senescence and the molecular hallmarks of Alzheimer's disease. If you look at a lot of the molecular hallmarks of Alzheimer's disease, such as an increase in DNA damage, inflammation, mitochondrial dysfunction, a lot of these overlap with the canonical inducers of senescence, such as DNA damage-induced senescence, inflammation-related signaling coming from senescence, or mitochondrial dysfunction-induced senescence. As Pierre mentioned yesterday, the brain is particularly susceptible to the canonical inducers of senescence. Because neurons are postmitotic, they lack specific capabilities of DNA repair that proliferative or dividing cells have. And so this gives them less capacity to respond to damage, or DNA damage particularly. Also, the brain consumes about 20% of your body's total oxygen, and that just increases the risk of potential oxidative stress and resultant damage in the brain. And so how can neuronal senescence be investigated in humans? We all know about the Nobel Prize winning discovery to develop induced pluripotent stem cells about 10 to 20 years ago now. And so what you can do is you can take a fibroblast or a skin cell and you can induce pluripotency. And then you can differentiate that pretty much any cell type, or a lot of different cell types in these days. But you can also differentiate neurons. There's another really cool method that was developed about like 10 years ago, kind of from Mario Swarnig's lab at Stanford, also Rusty Gage's lab down at the Salk Institute in San Diego, where you can take a skin cell and actually go directly to another cell type, skipping the stem cell state. And this is kind of called transdifferentiation or direct differentiation. And the pros about this is actually that when you go through an induced pluripotent stem cell state, you're rejuvenating the cell type. So you're basically looking at a fetal cell type response. If we're talking about aging, we kind of want to contextualize adult or older cell type responses. And sometimes when you rejuvenate the cell state, you're missing some of those age-related responses. So this is kind of a pro of the direct differentiation approach, and this is what we took and developed at the Buck. This is kind of a little more detail on how it works. These two factors are pioneer transcription factors that can open closed chromatin to change a cell's state, which is normally not possible because you want your cells to stay your specific cell type usually. But in a dish, we can activate these transcription factors for 3 weeks, and then we are able to get a transdifferentiated neuron. And we didn't develop this technique. This is technique, and this plasmid is all pioneered out of Rusty Gage's lab at the Salk. And as I mentioned, this maintains transcriptomic and, importantly, epigenetic signatures that are present in the fibroblast. And so what does this look like? So implementing this direct differentiation platform at the Buck, this is a dense fibroblast culture. At 0 days, the transcription factors haven't been activated yet. And then you can see at day 21, or 3 weeks after, you can see after activating these transcription factors, you get neuronal-like morphology. So these projections, kind of this triangular shape that is normal of more adult differentiated neurons. And so I think what's also really cool about this technique is you can take two different cell types and manipulate them in a dish. So right now it's really hard to take neurons from a human brain, and then it's even harder, or pretty much impossible, to take those neurons and then do something with them in a dish to test like different stress responses. What we can do is we can directly compare the fibroblasts to the neurons to compare cell type responses. And specifically, we can test senescence responses from the same donor. So now we're able to rule out these changes, like if you wanted to look at a neuron or a fibroblast, we can rule out the differences between looking at different donors. Now we have the same donor, two different cell types, one cell type that's actually really hard to manipulate in a dish. And so that's what I did. I won't be showing much data today, but I'll just be summarizing the results. And so what we did is we directly compared senescent fibroblasts to senescent neurons within the same donor. And this really hasn't been done before in a human context, in particular by inducing senescence through DNA damage. And so what we did is we induced DNA damage through radiation. This is a canonical inducer of senescence. And then we just stained for different senescence-associated proteins. And so what we saw is actually that the fibroblasts exhibit a P16-associated senescence phenotype, while the neurons exhibit a P21-associated senescence phenotype. And this is important. I'll go into it later, but P16 and P21 are two big arms that kind of control senescence responses and cell cycle arrest. And it's interesting because also neurons are already cell cycle arrested, so how might they induce these factors if they're already not in a proliferative state? And then also I listed some other markers here. These are kind of canonical markers that go up or down or go in different directions depending on the context, but usually lamin B1, which is a nuclear membrane protein, goes down in senescence context, and then nuclear area in senescence context and or nuclear size goes up in senescence context and with older age in a variety of different cell types. This was what we saw in the senescent neurons, as I mentioned, was only P21 going up and no changes in other canonical associated senescence proteins. And so what we saw here, just summarizing this, is that these two arms that control cell cycle arrest, usually in a canonical senescence response, P16 and P21, after DNA damage, it looks like the senescent neurons are pretty much only utilizing P21. When we looked at, we can also do RNA sequencing from these two different populations, and then we can perform some gene set enrichment analyses, and this is kind of to look at the SASP from RNA sequencing. Ideally, you would look at a protein level metric, and that's what we want to do next, but here we're just predicting what the SASP might look like from the genes. And so the predicted SASP in the fibroblasts actually went down, which is kind of against canonically what a lot of cell senescence models have shown in the literature. A lot of senescence models have used fibroblasts before, and they've actually shown this complex and heterogeneous SASP. But in these primary dermal fibroblasts after irradiation, we actually see predicted SASP enrichment to go down. And then with the senescent neurons, we actually see the SASP enrichment to go up. And so this is also against kind of some canonical ideas of neurons as well, because neurons are thought to be less secretory than other cell types in the brain, such as astrocytes or microglia. And just to summarize this, direct comparison between the senescent fibroblasts and senescent neurons, so we're seeing in the senescent fibroblasts kind of this more canonical senescent-like response: increase in P16, decrease in lamin B1, increase in nuclear size. The senescent neurons have more P21-associated senescence response. And then we also looked at the predicted SASP and then predicted transcription factors that might be driving the SASP. And it looks like they have an increase in SASP, and this might be driven by NF-kappa beta 1, which is a primary driver of the SASP in multiple different contexts. So why is this important? The novelty of our findings could really help develop therapeutics for Alzheimer's disease in the future. I know this is very basic research, but I'd really be happy to collaborate with other people on how we could move these RNA sequencing signatures to find novel senolytics to target a specific cell type or senescent cell type in the brain. I didn't show this point, but we also compared DNA repair dynamics between the fibroblasts and the neurons. It looks like the neurons actually have a decreased capacity for DNA repair. That's important because, as I mentioned earlier, they're post-mitotic. They have less capabilities for DNA repair. Potentially, you could provide in the future DNA repair enhancements, and this really is not very studied in the fields and in the aging field in general. Maybe you could push, instead of eliminating senescent cells, you could push to prevent senescent cell states through DNA repair modulations. But that's like a future aim of mine. Hopefully I want to do in a postdoc. The fact that, or how we're showing that senescent neurons adopt this predicted SAS, you could develop neuron-specific anti-inflammatory therapies. I know those are kind of out in terms of the clinic, in terms of targeting AD-specific related pathologies right now. But also this last point of how senescent neurons display a P21 associated senescence response This is really important to developing cell type and AD-specific senolytics. And this has been shown to be relevant in preclinical mouse models. They've given senolytics to older mice, shown elimination of senescent-like populations, and then also potential increases in neurogenesis. And so I think also this fact that there was no change in nuclear structure phenotypes could actually be a cool part of neurons, that maybe they're a little more protective than other cell types, and the fact that they might want to survive instead of die from a senescent stimuli compared to other cell types. And maybe you could understand these mechanisms to potentially develop drugs to maintain nuclear structure after DNA damage or something like that. I'm hoping to publish these results with my paper and my thesis work soon. We're really thankful for the organizers here. Hopefully we can use some of the award to push these papers into publication as soon as possible. The last thing I'll talk about really quickly is that we can also directly differentiate fibroblasts to other cell types. And so we can differentiate fibroblasts to oligodendrocytes. And this is a really understudied cell type in the brain. What these cell types do is they're crucial for myelinating axons, which allow for proper electrical signaling. It's kind of like wiring insulation on like a wire to allow for the propagation of a signal. And usually in a dish right now, when you're studying neurons, there's actually no myelination of the axons. So if you could develop this other platform and maybe co-culture these cell types together, oligodendrocytes, and neurons, you could have myelinated neurons in a dish, which would be a really cool model to have. But right now, this is a project that was developed from actually one of our research associates in collaboration with us and Lisa Ellerby at the Buck. And what they're doing is the same kind of direct differentiation platform, just with different transcription factors as earlier, and directly differentiating fibroblasts to oligodendrocytes. And so what this looks like, again, is this fibroblast culture here. And then 16 days after activating the transcription factors, you can see this oligodendrocyte-like morphology in a dish within about 2 weeks, which is really, really cool to see that underneath a dish under your own eyes. And what has been already observed in the literature is that with this induced oligodendrocyte platform from older patients, there are senescent markers that are present, such as P16, P21. And so what myself, Anya, and Lisa wanted to ask was, are these senescence markers present in induced oligodendrocytes from Alzheimer's disease patients? How relevant is this to Alzheimer's disease? And so this was kind of the platform, basically the same thing except taking healthy fibroblasts from healthy patients and then fibroblasts from AD patients, directly differentiating them into oligodendrocytes, and then doing bulk RNA sequencing, and then a gene set enrichment analysis. And so this is kind of a wheel of kind of the hallmarks of aging based on RNA sequencing signatures, but we actually don't see much cellular senescence signatures from this, but we see more of a chronic inflammation signature. And so this is something we're pursuing more, and then also we could use this model to induce DNA damage and see if this relates to senescent-like state in the brain, in particular in oligodendrocytes. And so just summarizing everything, I mean, really this is basic research, but this is hopefully going to help push the field forward to develop better therapeutics and for Alzheimer's disease in the future. I already mentioned the novel findings we have from the senescent neurons, but just putting here that we've also found some novel findings from these AD oligodendrocytes that they're displaying inflammatory signatures as well. And yeah. Thank you for listening. I really appreciate the opportunity being here, and happy to take questions.
All right, yes, if you put up your hands, yes, right. I'm going to ask you, and I'm going to ask you next. All right, thank you. So just 2 questions right now, because, again, we want to make sure we don't go over like we did yesterday. Here we go.
So, very interesting work. I wanted to know if you started studying existing therapeutics in the fibroblast studies, such as atorvastatin, which we know decreases NF-kappa B activation, because we do have a lot of existing therapeutics that affect those factors.
Thank you. That's a great question. I'm going to just bring it over to Brandon here. And here we go.
I haven't heard of that particular therapeutic, but I know there's a lot of different modulators of NF-kappa B, beta. I would definitely be open to testing that in our model, especially in the senescent neuron model. And Lipitor? Sorry, I'm just the basic research scientist, but
what she asked so that it could be on the stream.
oh,
Yeah, repeat.
she asked if I'm saying this correctly, if I've heard of Lipitor, which is an NF-kappa beta modulator, and particularly for fibroblasts.
No, it's a medication to control cholesterol that can also have an additional effect of decreasing activation of NF-kappa B. So many people feel that the statins not only are decreasing cholesterol, but they have a secondary effect of decreasing inflammation, particularly having this role in NF-kappa B. So I don't know if you tried to add, that drug to your studies to see if that affects the results. If you started testing therapeutics.
I have not.
Again, please.
Oh. She's saying that Lipitor has been used as a statin, and it's been shown to have other modulating effects other than cholesterol lowering, in particular to NF-kappa beta and inflammation. I mean, I've heard of the beneficial effects of statins, not just for cholesterol. I've heard of the beneficial effect of statins to give like earlier in life, not just when you have higher cholesterol. I have not particularly heard of the benefits, or I've not used this in my system yet, but I'd be open to it for sure. And maybe like looking into the systemic effects of statins, like in the brain, could be of interest. People have been on them for a while. Yeah.
Thanks so much. We got one more question. All right, here we go.
Yes, thank you. Fascinating presentation. I'm wondering if you're aware of the work of Dr. Jean Hébert, who is currently a program director at ARPA-H. He wrote a book called Replacing Aging, and the connection here is if this transdifferentiation from skin cells into neurons and oligodendrocytes can be achieved at scale for a given individual, then his approach of incrementally surgically replacing parts of the brain, say aged neurons with new neurons that are that patient's own neurons, could be implemented. So I'm curious as to your thoughts on this.
Okay, great question.
So he was asking about generally replacement therapies and if John Hébert has, or I've looked into like his research on replacing progressively like neurons or other cell types of the brain. Yes, I think our PhD program is really cool because we've had a lot of discussions on like primary theories of aging and how to reverse that. I think replacement and partial reprogramming are probably the two only therapies right now, potential therapies that could actually reverse aging. In terms of replacement, I think the toughest part would actually be that you'd have to have progressive surgeries, and each surgery is a huge inflammatory bout in your system, and I think it would be really dangerous to recommend at this point. I would say, if anything, I mean, I don't know if you'd want to use the direct differentiation platform because you're maintaining the age-related specific marks, and so maybe you want to use more of a platform that would rejuvenate the cell types and then progressively replace into the brain. But I think replacement is very, very far off, if I have to say that myself.
All right, thank you. Great questions and great talk, Brandon. Thank you so much. All right.